All right, thanks, Barry. What we're going to talk about today is with all the work we've been doing on PNCA, we've probably been working on it 3 or 4 years. And instead of getting up here, and I think we talked about it last year and talking about some of the old data, we're going to talk just very little bit about that and a lot of new data. So, and this data is so new, it's still just coming out of the lab. I'm getting data this week. So these slides are pretty fresh, so be gentle. There's going to be some data in there. It'll take me a minute to look at because we're still analyzing it. But I just want to, started looking at this talk and putting it together. It was 110 slides to begin with. I was able to get it down to 49, so that's as few as I can do. So we'll get going. So we started this and we called it the never-ending experiment. We started and it went on and on and on and on. So we said, okay, just like any mutation PNCA, you keep looking, you find more and more and more. So the new thing is the never-ending story of PNCA, because I wish I could tell you today we're finished, but we're not, and you're going to see why we're not. I think there's still going to take some more work, and probably another PZA talk again next year. So when we started doing PZA work, we're looking, most times we follow a strategy is using the phenotypic DST for a drug to help develop molecular assays. Problem with that is very difficult, especially when the phenotypic DST is not as reproducible as you would hope or reliable, so you can't really go in that direction. What we thought with PNCA instead, we would use more of a molecular assay, in this case sequencing, to help us work back to the phenotypic DST and maybe hopefully improve that. Well, that wasn't enough, so we developed some more tools, and we use enzymatic assays. We can run 2 different ones, either in H37 or TB, using something like the Wayne assay and stuff. But we also developed the E. coli one I'll talk about. And we use that to help define both phenotypic and molecular assays at the same time. So we took like a multi-pronged approach to try to tackle this problem. And when we started, we were like, this will only take about a year and we'll be done. Well, we were wrong. Many of you know the problems with PZA, especially with PZA testing. You know it requires an acidic culture. Problem is, if you get a too alkaline, then the pyrazinamide is not going to convert it. You get a too acidic, then you're going to start, some of the bugs aren't going to grow. So you have to find a fine balance in type with the pH of the media. PZA is active against all M. tuberculosis complex except for bovis, which actually has a natural occurring polymorphism inside of polymorphism PNCA, which disrupts it at one of the key residues involved in the metal binding site. Some of the big factors that do affect PZA testing, as I mentioned before, is the pH of the media. There have been plenty of studies looking at that, and we were just talking a while ago about how, putting pyrazinamide in something like a Trek-like plate. The problem is that media has to be different than the rest of the plate, and getting it under the right conditions where you get reproducible results is very difficult. Another thing that we noticed too, and many have, is the inoculation size can really affect the your PZA results. And what we wanted to focus on initially was not the pH, because that's pretty standard now in the MGIT system, but looking at the inoculation size. Can we kind of modify the current protocol in a way where we can make it more reproducible and using all the fully characterized strains in the development of that? So in the phase 1 part of the study, we had already done a molecular drug resistance survey, so we had a lot of strains where we'd done Sanger sequencing and looking at PNCA. So we picked from that collection of strains. We had 149 non-synonymous SNPs. 98 of these were unique and affecting 68 different codons. We also have 23 strains that had indels or promoter mutations. The standard MGIT protocol basically, put my glasses on. There we go. As we always want, depending on the day you catch it post-positive, we concentrated on day 1 and 2 to keep it standardized. In reality, that's kind of hard to do in a clinical setting, but we wanted to keep this as close as we could to everything same across. You typically, if you catch it 1 to 2 days post-positive, you do a 1 to 10 dilution of your no drug control for your inoculation, and then there's no dilution to going into the PZA. So there's a ratio of 10 to 1 number of cells, and you're either no drug versus the PZA. What we decided to do was to bump that up another tenfold. So what we did was dilute our no drug control 1 to 100 and with our PZA 1 to 10, and we compared that across a bunch of subset of strains and saw that it was reproducible, and we used this test moving forward in our phase 1 assay. Just to kind of summarize those results, if you look at the MICs for these various SNPs, you'll see a lot of them had an MIC greater than or equal to 800 micrograms per mil. That was affecting 85 strains, 57% of them, and affected 42 different codons. Then we had some that were less than equal to 300, and then we had some 400 to 600, and then we had some we call you pick one. We test the strain 4 or 5 times, and we get 4 or 5 different answers. So, and these are some of the problematic mutations. Their MICs are sometimes near the critical concentration, but we were getting fluctuations from 100 to 600, and trying to control the inoculation as careful as possible. Some of the codons that where we did have problems was like codon 12 and aspartic acid alanine. It had very highly variable MICs between isolates. And this is because you had either alanine or glutamic acid, and depending on which mutation you had, it affected the MIC itself. Codon 47, I'll go in a little bit more detail now, and that's the W strain, by the way, Barry. And codon 103 was a tyrosine to histidine. We had 3 of those, but you can see one isolate tested at greater than 800, one isolate tested less than equal to 300 3 times, and one isolate was all over the place. We didn't have whole genome sequence at the time on these strains to see what other mutations they had and how it reversed with these 3 strains. Or, you know, a lot of times you see PNCA mutation that's the same mutation is usually a good clonal marker. But codon 103 tyrosine to histidine is a very more of a common mutation, if you can say that for any PNCA mutation. But in codon 47, depending on the mutation you had, if you had the threonine to proline, the MIC was pretty stable, greater than 800. But the threonine to alanine, which is the marker for the W strain that was circulating in the states of drug-resistant strain, his MIC was less than equal to 300. So we had to do another MIC panel, and his MIC was always between 100 to 200. And this is why early on, when they were looking at this strain, some labs would get it resistant, some labs would get it susceptible, and it was never really reproducible, and it was because the MIC is very close to that critical concentration. This is some work we published actually with Rob Warren's group. We had 5 mutations, and he had, I think, 5 in his collection. We had a ton of data on these, enzymatic data, MIC data, and what we were able to show, not all mutations in PNC actually confer resistance. I haven't really found any more in our collection, but we haven't really been looking at them hard right now because we're concentrating on something else, and I don't think you found out any more other, have you, Rob? Okay, so we're at 10 well-known characterized strains, mutations that we know don't confer resistance. There also, you have to worry about synonymous mutations. Serine, 65 serine is one of the most common ones. We've seen another 3 or 4 other ones, but I mean, typically synonymous mutation PNCA are fairly rare. MICs for indels and promoters, I mean, this is pretty straightforward. They typically are always really high. So if you have a frame shift or anything or a promoter mutation that's defined, you pretty much are always going to test resistant. We also looked at MICs for wild type because we kind of biased that data because we looked at all PNCA mutations. So we had to go back and look at a lot of wild type PNCA strains and look at these, especially the inoculation, and see how that affected. And again, we did the standard protocol and what we call the experimental protocol. So when we did this, we basically saw that most of them were consistent. And what we did see is, here, I'll get into this, is if you looked at the standard protocol and compared it to the experimental, if they tested susceptible in both, it was about 72% of the strains. We did have some that were testing resistant to resistant, and when we went back and looked at that, we thought we had some little bit of heteroresistance. They weren't really truly wild type. The ones that went from resistant to susceptible, those were sequenced. Those were definitely wild type, and what we see was with the lower inoculum, we are getting now what we consider not false positive. And also, we never really had any went from susceptible to resistant using the experimental method. So based on all the data where we tested hundreds of strains using this experimental method, we did notice that we were having some timeouts because the 1 to 10 dilution and 1 to 100 on the no growth control, before you reach 13, 14 days, if you don't get growth in there, it will time out. So what we also want to do is do a multi-site center evaluation to say, does the reduced inoculum really have any effect, and is it reproducible interlaboratory and intralaboratory? So working with APHL, Glenn Morlock from my team and Francis Tyrell from the laboratory capacity team put together our RFP to look at that, and we picked basically, going into this study, the hypothesis was that over-inoculation causes false resistance, and there's poor reproducibility. We thought the lower inoculum would help solve some of these problems, and then we didn't see if it was going to be universal across a well-characterized set of strains. So we did the 3 inoculation schemes now, which I'll go over to in a minute. We did 10 laboratories, 9 extramural, and 1 we did my team participated too. We did 20 isolates, 15 were unique, and 5 were duplicates of those. And we had them run the assay in duplicates as well. So the BD method is one I just showed you. They're catching on 1 day, 1 and 2, standard protocol. We did 2 different types of dilutions, either a 1 to 25 for the no growth and 1 to 2.5 for the PZA, and we did a 1 to 50 and also a 1 to 5. So instead of doing the 1 to 10, we cut that to 1 to 5 and 1 to 2.5. And this is just the workflow that they had to follow and in this assay. So basically what we did was look at the phenotypic and MIC data of all the set of strains we had, and also the genotypic where we had Sanger and whole genome sequencing, and pulled a set of strains. We had these, we had some that were very consistent with their PZA results over all the times we tested them, and we also had some that were highly variable, so we considered to test those as well. We had some that were always high-level resistance, which no lab should miss that, and it should, no matter which inoculation you're using, should test resistant. We had some that were consistently PZA susceptible with very low MICs, and we had 9 strains that were very variable with the MICs, depending on how the testing was done. Basically, they did all the tests in duplicate, and we ended up with 1,200 tests total across all the different sites, and then we did categorical resistance susceptible from that. I'm not going to go over all the data. This is some of the data we were collecting, though. We're looking at the agreement between duplicates within a lab and across the laboratories, and looking at percent agreement. All that would look pretty good. We did have one lab that kind of stood out, and their inter-laboratory reproducibility was a little off. We thought it was some transcription errors, but we haven't been able to show that, so I'm not sure what's happening there. Basically, some of the take-home message here, I won't go over the data, is basically with the PNCA with susceptibility, it seems to be good. The strains are really susceptible or susceptible. But what we did notice is there's some strains that are kind of not as reproducible, but what we did notice is the lower the inoculation tended to give you what our expected result. And these were not timing out at 1 to 2.5 or 1 to 5. So as we went down lower in the inoculation, our accuracy and precision actually increased. We did have 3 unique mutations. Again, we used some of the ones that we know that didn't confer resistance, and again, this proved, again, we can keep the paper going, that they definitely don't confer resistance. It was nice that 9 of the labs tested them, they were susceptible also. When you look at high-level resistance, again, the inoculation didn't seem to have an effect. If their MICs are greater than 800, that's not going to affect it at all, and that's what we saw. Basically, if you look at guys who were inconclusive, again, what the trend was across the sites that the lower inoculation tended to improve the accuracy somewhat. But, is this the correct results? Because are we biased in it? Are we doing the dilution to get the results that we expect? What does this mean clinically? We have enzymology data. We've done a lot of that. I mean, do we need to do animal testing on some of these strains too? So it's still questions. Are we biased in what we're doing based on pre-expected results? So from this, we learned that it's kind of like with all the TB, it's almost like a Goldilocks thing. Too much is too bad, too little is too bad, and you just got to get it just right. We know that can be very difficult to achieve an ideal inoculation, especially when you can catch things at 1 or 2 days post-positive versus 3 or 4. So it still looks like, with PZA, the reduced inoculation did help. We noticed that strains respond differently to the inoculation. Highly susceptible, highly resistant didn't seem to really matter. But when you had things that were in that realm of being susceptible or resistant on their MIC, that the lower inoculation tended to improve that somewhat. So now with having the DST and what we call the revised inoculation scheme, the other part of this was an enzymatic assay instead of having to do the Wayne assay on everything. So what we did was use E. coli. We found a strain where PNCA had been deleted from E. coli, and then we cloned back in every mutation that we had, and we compared that back to the data. And the way this works is you clone PNCA to the E. coli, you can induce it with IPTGs, an inducible promoter, in the presence of pyrazinamide. You can add an iron substrate, and what now you're looking for, do you get a color change or not? If you get no color change, we're assuming that you have a non-functional pyrazinamidase or it's not making it at all, and that you would be resistant. Well, if you get our color changes in this case, then you would assume that the pyrazinamide is active and you would be susceptible. So, and then we had it's not a quantitative, it's more of a qualitative grading scale from 0 to 4, depending on which tubes turned red and which ones did not. And then you can make this as the grading scale, and the higher the activity, the higher the MIC, the lower lower the activity of the enzyme, the higher the MIC, and vice versa. So what we also had to do was to develop an antibody, because if you had negative data, you want to make sure at least the protein was being made, or was it unstable? And we have made a peptide antibody that we could actually do that. And we, depending on what the mutations were, though, the antibody most of the time would work unless it deleted that region. So always having something. So if you look at the strains here with MIC data, this is a little bit out of date. Now we have done all of those strains in our collection. And when we try to grade this system out, comparing an enzyme grade of 0 or 1, and then calculating against the MIC for 100 or greater being resistant, you can calculate your sensitivity specificity, and our sensitivity in this case was about 95%. So it looks like the assay correlated really well in this phase of the study. So if you look at PNC, as Barry mentioned, mutations can be found everywhere. So 187 codons, and just remember green is wild type. You start looking at the gene, though, and you start seeing from other people's our collection and then what's in the database, you start seeing a list of mutations. And there's all the yellow is the codons where we saw mutations. You had another database, you had the orange, you had another database, you get the purple, and I just got tired of doing this, so we stopped. Because it was like every week you find a new mutation. And the problem is, one codon can change to 5 or 6 other codons. So it's just, it's unlimited the kind of mutations you can get here. So I'm going to skip over there. So instead of doing this way, we decided, okay, fine, most of the changes are due to a single nucleotide change, so let's convert every nucleotide to the other 3 nucleotide and see if it's synonymous or non-synonymous, and if it changes the amino acid, then we're going to make that. So we did that, and we had 1,041 to make. I can tell you we're done. We're still cleaning up the data, but when I go back and look at every mutation we made and look, there's 4 or 5 databases we use. We use our internal database, we use the Broad sequencing database, we use TB Dream database, we use Paulo and Sven had sent me a long list of strains from Europe, and Fermaz, graduate student, had published their systematic review paper with all the any mutation found, and when we do that, about 400 of the ones we made are found in one of those databases. So the other 600 or 700 are probably yet to be found, and they probably do exist somewhere. I'm not going to tell you all the numbers now because a lot of them didn't, very conservative changes did not have any effect. We're looking at that data with our categorical data. Is there 100% agreement? No, it's not. So there are quite a bit of discordant results, and what do those mean? We don't know yet, so we've got to continue to work on this project, and maybe have to do some functional genetics and stuff. And getting to that, basically we do have discordant results like I mentioned. We need to do some functional genetics, and we're not going to do 1,041 functional genetic studies in TB. We're designed in a way where we can do this pretty rapid, but it's going to take some time. We're just going to kind of hand select quite a few of these to look at and make these point mutations in a good, clean genetic background. We're creating the database with all these mutations, and we're going to try to collate it with everything else is in the literature. And the other thing is with some of the, in our hands, we didn't see a lot of PNCA wild type that were truly, truly resistant, but see some in our clinical testing. So the question, what is the role of the other genes? What is the role of RSPA and PAN-D and things like that? Problem is when I look in the 2,000 or 3,000 strains that we've sequenced and say, what are some of the polymorphisms we're already seeing in these genes? In RSPA, we already got 53 different alleles, and there's very little resistance in our database. So there's going to be a lot of polymorphisms within some of these genes, and what do they mean, and how do you rule out what's natural killer polymorphism versus what causes drug resistance? You look at PAN-D, on the other hand, there's only 10 different alleles. So we can go back and look through the data and see what that really means, and then going into Resect TB will help too, because now we'll have 5,000, 6,000 strains, and a lot of those are resistant strains, 30-something%. So we'll be able to see what kind of mutations are circulating and are they lineage markers or not. So this work has been going on, like I said, 3 or 4 years. And a lot of people, almost my whole team, has been working on it. We call them the PZA team. A lot of people have come and gone, but it's been a tremendous amount of work that these guys have done. We're continuing to do more work, and maybe one day we'll get up here and say we know everything about PZA and PNCA, but I doubt it. So, thank you.
Since it doesn't look like we're having a panel, since there's no chairs, if there's any questions for Jamie, we might as well take them now.
Well, this is very impressive, Jimmy, I must say. This is a lot of work. So I was just wondering, your 3 mutations you mentioned, which did not confer resistance, still susceptible, whether those 3 mutations, whether you'll be able to express and look at the enzyme activity, whether the 3 mutations have any effect on the enzymatic activity at all?
Qualitatively, no. All of it looked just like wild-type levels, but again, that's a qualitative assay. We haven't done quantitative or specific activity, but from what we can tell in the MIC of the strains, that they really had minimal effect on the activity of the enzyme.
Thanks for this talk. I think you mentioned it very briefly, but did you see any interactions between different point mutations within the PNCA gene at all?
Typically, in PNCA, you typically only have 1 mutation, and so they're all, it's not like you usually have multiple mutations within the gene. We do have some isolates that have more than 1. A lot of times you'll have a silent mutation at serine 65, and then another one, but I would say more than 97, 98% of our strains that have a PNCA mutation is just 1 single mutation. It's usually not a double mutation, like in 2 different codons.